So the other area we work, so that's the food side and the types of things we're doing in the food area. The other area is the rare sugars for pharmaceutical applications. And it really is an offshoot of what we've been doing in the food side, in that we can then take things like polyols and other inexpensive carbohydrates and make them into higher value added products. So all of the technology I just talked about and the enzymes that we use can either be turned on and off to make different types of rare sugars, or the products, the xylitol and the mannitol and other polyols we've worked on, can then be converted in one step to a variety of rare sugars. We also have enzymes for making sugar 1-phosphates and activated sugars, and I'll talk about what those are used for in a little bit. These are basically all the building blocks for attaching sugars to other molecules, and I'll talk a little bit why that's important. But rare sugars, these single sugars, are important in a number of drugs. This slide's a little bit old, but you can see there's a number of drugs on the market or going through clinicals that have as their core rare sugars like L-ribose, L-gulose, L-galactose. They can be synthesized from those molecules. The trouble is those compounds are extremely expensive, and some rare sugars basically you can't purchase. You can't purchase them anywhere. And in fact, if you look, here's a bunch of sugars we can make. If you were to go to Sigma, which is a chemical supplier, and buy 100 grams, which is like a salt shaker full, these would cost you anywhere from $100,000 to some of these aren't even available. So the goal here is to reduce to be able to make these affordable, so researchers can use them and they can be developed into new drugs. So a lot of the opportunity for sugars, people haven't even been able to test these as building blocks for new drugs. So we're opening up a whole new branch of chemistry and research for the pharmaceutical companies by being able to supply these types of chemicals. And here's kind of why how we work, why our technology is good. L-ribose, which is a key intermediate for a number of drugs. Right now, the main process, it is one that can be made, but there's a 3-step process that's currently being used to produce it, and each of those are very low yields. So at the end, you have a very low yield of L-ribose from a cheap starting material, but it's still very low yield. Whereas we can take a single product and get an 81% yield. Actually, it's over 85% now in a single step. And how did we develop that? Well, we found some unique enzymes. We've got a dehydrogenase that can convert a polyol into a rare sugar. It's a proprietary enzyme that we've then developed mutants of so that they can do this reaction at very high yield and very high productivity. So for instance, this enzyme and the conversion of ribitol into L-ribose, the wild type strain, for instance, produces L-ribose at 2 grams per, about 2-3 grams per liter hour. After 2 rounds, after carrying out some directed evolution mutagenesis on this, we were able to increase that to about 12 grams per liter hour. We can produce, and here's the wild type production, here's the mutant production. And it can not only can we produce L-ribose, but this enzyme can be used to produce a whole variety of different rare sugars, L-gulose, L-galactose, L-D-lixose. These are all sugars that are not generally found in nature, are very hard to get, very difficult to get, and we can now produce at fairly high yields.